Today, we're going to be talking about this paper, which I know I'm hitting you guys in the beginning of the year with a lot of really hard stuff. It will actually get easier in this class as we go through the year. Because right now we're going to be talking about framing real science experiments. What does a real science experiment look like, right? Because I feel like a lot of times in high school science, in middle school science, and even some college science, you don't get a real idea of what scientists are actually doing, right? You have these stupid experiments. I say stupid because we already know what the result's going to be, right? And you just keep doing them to learn a content or to learn something else. So I want to talk about how real scientists frame their learning and kind of about the process to hopefully get you appreciating what scientists are doing a little bit more, and also maybe connecting that with experiments and whatnot and seeing the relationship between why we do experiments the way we do in high school and how that could translate into becoming a scientist. So let's have everyone type a 1 in the chat if you were able to check out this article beforehand. So you looked at the article, you watched the video possibly of me reading the article. Well, if you're saying which article, that's probably not a good sign. I will give you a link to it right now. So this is the article, which we'll also look at. So if you have not checked out the article yet, then I would be taking notes if I were you personally, because we are going to go through the answers to the questions, and we're going to talk about what this article is about, right? But I see 3 people gave a 1 here saying that they were able to check out this article before. I'm going to guess you are the 3 people who've already turned in your pre-work, right? So that's awesome. So let's talk a little bit about that, how you're going to be graded for this session today. So in modules, I warned you last week during the virtual session that this one did have pre-work, right? And it had something for you to do in advance. There will be other sessions like that as well. In fact, the next virtual session, there's no pre-work. So that's a good news for some of you. If you checked out here, lesson 4, how can we use our knowledge of science and statistics to understand what is currently going on in science research? And here it says, Socratic Seminar, check me out to see the instructions and questions. So hopefully some of you checked this out already. I know at least 3 people did. 3 out of 9. We got a third people checking it out in advance. But you should have seen that these are the 7 questions that you were looking to answer through looking through the article on your own or hearing me read and explain the article. So why is this study important? What are some of the findings from this study? What background information is needed to understand this study? Describe one way they used math. Why is hospital pneumonia so bad? What is one way that reading this paper has strengthened your understanding of science? And then what was the hardest part about reading this paper? Right, so some of these questions were your opinion, and then some of them were finding things or explaining things from the article. So in order to get credit for today, you're going to have to turn in your pre-work, which is answers to these 7 questions. And I will be grading you on this rubric. So I will be giving you 15 points if you answered the 7 questions to the best of your ability, right? Even if that's saying, I think it's this, I'm not sure, that's still trying to do the pre-work. If you respond to at least one of these questions in the discussion today, you'll also be getting 10 points. And then if you want to get a full 30 out of 30, you're going to respond to a classmate, right? So perhaps Alexis says that she thought this article, the hardest part was the vocab, right? Maybe Joelle will jump in and say, Alexis, I agree with you. Some of those words were insane. I didn't even know how to pronounce them. And I could tell that Miss Jamie didn't know how to pronounce them either, right? Because if you listen to me read to you guys, some of the words, even I'm like, I don't know how to pronounce this. So Bonnie's saying I had a hard time because I've been in the hospital. Yeah, right? So if you had other stuff going on, this was not an easy thing to quickly do to understand this article, right? So we'll probably have some discussion around that as well. So that's how you're going to be graded today. So if you just turn in the questions, you'll get 50% of the grade, right? So try to make sure that you respond. Oh, in the past, yeah. Okay, yeah, well, I'm glad you weren't in the hospital recently, Bonnie. So try to respond to one of the questions, even if your response is your opinion, right? You're not quite sure, maybe it's a question, etc. That counts as responding. And I will give you guys a little heads up towards the end of how many people said something, right? So you know where you are if you've spoken already or not. So we're going to start off, since we only had 3 people out of our 9 people that are here, we're going to start off going through the PowerPoint just pretty quickly that you should have gone through on your own and that I encourage you to go through after this session. So what you should have gotten was that this is a real science paper, and I say that literally to mean real science papers are not written for the public, which you probably noticed, right? This is not BuzzFeed, right? They're not breaking down science for you. This was real advanced graduate school work. Of course, we are going to try to draw some conclusions on this, and we're going to look for clues about the testing, right? Because no good science experiment exists without math, unfortunately. Or fortunately, if you like math, then awesome. So here was a video of me reading this article, which I actually noticed. Okay, there we go. I was going to say a few people may have had issues getting it to load. Notice it's 45 minutes long, so we're not going to watch that right now. That would be our whole session time. But I do go through the PowerPoint and I read the parts of the article to you that I want you to understand. So you can just put that on and look at the article, right, and kind of hear what I'm saying and process. But really, what we're going to do is we're going to talk about first the title. So I can't even see the title because my little, whatchamacallit, my Zoom bar is in the way. But the science paper is called, which I'm going to have to escape to be able to read, Natural Killer Cell Intrinsic Type 1 IFN Signaling Controls Klebsiella pneumoniae Growth During Lung Infection. Now, you may have read the title and been like, peace out, I will not be reading this article, right? Which honestly, if you felt like that, that's okay. That's a normal feeling. Mr. Ling is in the room and he just made a face where he was like, what words were those? Right, so we're going to start breaking this stuff down. Well, first of all, we're going to want to know what natural killer cells are, right? That sounds like something that we probably need to know. We're going to want to know what type 1 IFN signaling is. And then we may or may not already know what pneumonia is, but Klebsiella, yeah, I think that's how you say it, pneumoniae, is a specific type of pneumonia that impacts people who are already in the hospital. So here's an easy opportunity to get 10 points right now for participating. You can either speak out loud or say in the chat by typing, why do you think that hospital pneumonia is potentially more dangerous than regular pneumonia? And if you're not quite sure what pneumonia is, right, that's a lung infection where it's really difficult to breathe. People can die from it, right? It can kill you. And usually you have to take antibiotics and be monitored, right? So why would hospital pneumonia be something that is a pretty bad thing? Okay, we got a lot of answers. Awesome. So Abigail says, because of all the other sick people. Yes, right? So if somebody next to you has pneumonia and you're already sick in the hospital, well, what are you going to do? You can't move away from them, right? You guys know hospitals are crowded. Colin says the people in the hospital are already weak. Their immune system might not be able to fight it off. Exactly, right? You're usually in the hospital because there's something wrong with your body, right? A weak immune systems in particular can be really, really bad. It's more dangerous because they're in such an enclosed and dense space. Good, it's easy for it to spread. They're already sick, their immune system can't fight it, and more dangerous because people could get more ill. Yeah, right? So exactly. Really, the thing is, you're already sick, and now pneumonia could be something that infects you and then kills you, right? There's another type of disease in hospitals that's pretty common, another pathogen, another bacteria that you might have heard of, a staph infection, right? It's a similar concept. Your immune system's already weakened, or you're already sick, you're already working overdrive to try to fight whatever you have. It's super easy to get another infection on top of that. My mom had cancer when I was little, and I remember that we had to be really careful in making sure that she didn't get sick. Exactly, right? So especially in certain parts of hospitals, they won't even let you see people if you're sick, right? Because they don't want you to spread it. And again, all for reasons that you guys have already mentioned here. So I'm just going to write down who spoke here just so that I can keep track of this. We've got Abigail, we've got Colin, we've got Bonnie, we've got Georgia, we've got Joelle, we've got Briley, and I think that was everybody. Yeah. Get sick, you need oxygen. Yeah. Okay, so the first thing really that we want to mention with this paper is that these scientists are trying to solve a problem that is pretty important, right? A pneumonia strain that affects people in the hospital already. It's called hospital pneumonia in common language. So a little bit of background, right? We will talk more about the immune system in second semester of biology. So if that's something that interests you, we will talk about that a little bit more in the future. But really, your immune system is designed to kill bacteria and viruses, right? That's what its purpose is. There are a lot of cells. In fact, you can take an entire full year course on the immune system and still probably only know part of it because there's so much going on. But the two types of cells that you really need to understand for this particular article are the macrophage and the natural killer cell. Right, natural killer cells look pretty gross. That's one right there, NK. It kind of looks diseased, really. But natural killer cells actually can destroy your own personal cells. And macrophages sort of behave like a Pac-Man. So if you went through this PowerPoint already, you already saw this, right? But you can think of a macrophage, also known as a white blood cell, right? They target everything. They kill bacteria, viruses, fungi, and parasites, right? So no matter what kind of pathogen you get inside your body, macrophages are able to kill it. What do? They eat cells, right? And it's literally, I mean, this, you might seem like, oh, a Pac-Man, haha. No. This is actually what it looks like, right? They would eat a bacteria cell or a parasite or fungi or whatever it is, and then they internally destroy it. Right? So they're pretty important, and you guys may have even heard of white blood cells before. If you have low white blood cell count, right, that's not good. That means you don't have a lot of these macrophages to kill things. Natural killer cells, on the other hand, they live in your blood as well, right? And they are special because they can kill your own cells that have been infected. So they can tell the difference between a healthy cell inside your body and an infected cell. So that takes a little bit of recognition, right? You don't want natural killer cells killing cells that are normal and healthy in your body. Sometimes that happens, right? That can result in autoimmune diseases. You may have heard of autoimmune diseases where your body's attacking itself, right? But usually they don't do that. So they can destroy tumor cells. They can also destroy cells that have been infected with a bacteria or a virus. So they sort of just shoot a cell with signals that basically tell it to die. So it's almost like my immune system teacher, when I took an immune class, right, she was basically like, imagine it's like they have a machine gun and they just shoot at cells that need to die, and then those cells die. So it sounds kind of like a video game, and it sort of is, right? It's happening inside your body. But the natural killer cells are super important, and they weren't really well understood until pretty recently. Interferon, another thing from the title, right, is a signaling molecule that's released by host cells in response to bacteria. So it's kind of like a warning sign. So it's like if you are about to die, right, as a cell, you send out this signal to other cells saying, hey, I'm infected, I'm going to die, be careful, right? Which is kind of great, except if somebody next to you is infected and you're a cell, well, you're probably about to be infected too, or you might already be infected. So these signals need to be sent out really quickly, and they actually help the natural killer cells know which cells to kill. If you're sending out a signal that you've been infected, you're basically saying, kill me, please, natural killer cells, so that the body does not die, right? Sacrificing yourself as a cell for the greater good of the organism. Happens all the time in your body, right? In fact, cell death is a normal part of your body. right? All the cells in between your fingers had to die so that you don't have webbed hands, for example. So the interferon is just a signal. It's just a fancy word for a, SOS signal, essentially. So in this article, these are the main key points that you should have gotten. We will talk about these really quickly. Then we're going to try to answer the 7 pre-work questions, one of which we already answered about the hospital pneumonia. So the K pneumoniae, right, which is how it's abbreviated when you saw it in the paper, potentially if you looked at it, is becoming resistant to all known antibiotics. So no one really mentioned this before, but not only is hospital pneumonia dangerous because it affects people who are already in the hospital, but it's getting harder to kill with antibiotics, right? So you may have to take 2, 3, 4 rounds of antibiotics to kill this particular strain of pneumonia. It will eventually get to the point where we can't kill it, right? Where if you get pneumonia, it would be essentially a death sentence, right? Most likely you will die. You might survive, but the chances are pretty low, especially if you're already sick. So this antibiotic resistance, this is a huge current problem in medicine right now. Other things are becoming resistant to antibiotics as well, not just pneumonia, right? For example, gonorrhea, which is an STD, is becoming resistant to antibiotics. It could cause infertility, right? So it can have big implications on people, especially if they want to have kids, right? Also tuberculosis, which is another lung infection, becoming resistant to antibiotics. There's certain strains that literally cannot be killed anymore. We will talk in second semester about that in great detail because it's scary, right? And it's something that's happening right now. But basically, these scientists said, oh, wow, this pneumonia is not getting killed by antibiotics anymore. This is a problem, right? What else can we do? How can we get rid of it? So they started investigating this natural signal in your body, the interferon, right? Which is the signal that tells your body, I'm an infected cell, please help me. Really, please kill me. And they studied, well, is that interferon signal actually what is telling the macrophages and the natural killer cells to attack? Right? Really, what they're suggesting here, guys, this was a huge, huge deal. I believe this was from 2017, so last year. They're saying, well, how about instead of taking a medicine, you boost your body's own ability to destroy an infection? So what if you could get a shot that boosted your ability to fight an infection, right? That's huge. That's amazing, because how would your body be able to become antibiotic resistant? It won't, right? Your body will always adapt to whatever the pathogens are doing because you're evolving and so are they, right? So it's sort of this new idea. Yeah, it's like immune system steroids. That's a great way to put it, Georgia, where basically you're saying, oh, you're sick? Here, give your immune system some extra support, right? You guys might have heard there's like over-the-counter versions of this, right? Like take vitamin C, take zinc, right, to boost your immune system. It's the same concept, except this is on a molecular level with real molecules that in theory could actually help more than vitamin C would, right? If you took vitamin C and you had pneumonia, probably not doing much for you. So it also was found out in this study, interferons, they used to think were just from viruses, right? And viruses are already really hard. A lot of you guys know you can't treat viruses with antibiotics, right? They're a lot harder to get rid of. Eventually your body gets rid of them, but you might be sick for a long time, right? But then they found out, wait a second, bacteria infections release interferons too. This is a big deal, right? That was like a huge science discovery, essentially. So not only are they saying, why don't we use our own bodies? They're saying interferons are an important molecule that are released in many situations, not just from a virus. So let's talk about the questions that you're supposed to answer, which I sort of erased. By the way, if you didn't read the article yet, right now I would be jotting down notes about this. So I kind of gave you the answers to some of these things, but now you guys are going to articulate those answers back to me. Remember, if you participate, you will be getting 10 points. So we already got a lot of participation. Why is this study important? So based on all the things I just told you, / your own learning, listening to me read the article potentially, what would you say is the reason that this study is important? Good. Joelle says it's important because now we know the problem with this disease, right? Which is that antibiotics are not killing it anymore. We know they don't kill it anymore. We know why that is. And so now they're trying to find a new way to treat it. We can get our own body to fight diseases that we can't fight with current antibiotics. Excellent articulation there by Colin. It's important because it gives us insight to how our body works and can tell us how to treat illnesses. I love it, Bonnie. The body's healing itself, right? So for the past 100 years, really, not even 100, close to 100, since the invention of antibiotics, right, which was in World War II, World War I era. I believe they were invented in World War I, but didn't really come into mainstream until World War II. We really haven't had that many years of antibiotics, but the concept before was wait till somebody gets sick, give them an antibiotic, and then they heal, and then they're done. Now they're saying, what about using your body to figure out how to heal, right? It makes sense. Your body is designed to fight stuff. Okay, what are some of the findings from this study? Yeah, George was right. Back in the 18th century, if you got a fever, you were basically done for. Yeah, if you had strep throat, goodbye, right? You weren't surviving that without antibiotics. Maybe 1% of people would. If you got tuberculosis, you would die a slow, painful death where you coughed yourself to death, right? So yeah, it wasn't a pretty time, and we definitely don't want to go back to that. Antibiotics do not give you immunity. That is a great point / question from Joelle there. They don't actually, right? So when you take an antibiotic, it just kills the bacteria. You don't actually become immune to that disease, right? You're talking about a vaccine. So you've probably gotten vaccines, right? Vaccines are building your immunity so that you can fight off a virus. So there is a slight difference there. Yeah. But so yeah, antibiotics don't build your immunity. That means you could get hospital pneumonia 75 times in your life. Bonnie's right. It also kills your good bacteria, right? So if your body is healing itself, you wouldn't be killing your own good bacteria, like in your intestines, right? You wouldn't kill all that bacteria. Thank you, Colin, for bringing us back on track. Some of the findings from the study are that the cells can know to attack viruses and the disease is immune to antibiotics. Yeah, exactly, right? So interferons are released from viruses and from bacteria, right? And that our own cells, particularly macrophages and natural killer cells, will attack cells that are releasing interferons. Good. Briley just summarized that perfectly, right? Some findings from this study is that interferons not only work with viruses, but also work with bacteria, right? So interferons are released in multiple situations. That was a big conclusion here. To you guys, it probably doesn't seem like that big of a deal, right? You're like, all right, more sciencey stuff, whatever. But to the science community, it was like, oh, whoa, somebody figured out something cool here that we might be able to use. Yeah, that our body could heal itself, right? If maybe given a little bit of a boost or the right molecule to signal to the body, then yes, we could heal ourselves from inside out. Now, the hard thing is, this is going to be kind of a side question, because we're going to skip the background one, right? Because we kind of talked about it already. But how would we test this? Like, what would be the next step for this, do you guys think? We know this stuff now, right, from their mice experiments. What would we have to do to be able to use this as medicine? Yeah, so Joelle's right. We would have to test this on people, right? So how would you feel if you were in the hospital and you had pneumonia on top of whatever other reason you were in the hospital, and a doctor comes in and they say, we want to do this experimental test. We're not sure if it's going to work, and we're not going to give you antibiotics. right? Or maybe we've tried to give antibiotics and they didn't work. Yeah, so Colin makes a good point here. It's better than dying. Yeah, so if they tried antibiotics and they didn't work, I would say give me whatever you want, right? Let's see what we can do. However, it's going to be not a lot of people probably who are going to take that risk, right? Because of course you have to get the patient's consent. You can't just go in and start injecting people with stuff. Not in the real world, at least, right? So it would be really hard. This could take 10 plus years before it even became, something that was actually thought about being used. And that's part of what's hard with medicine and humans, right? Is that you can't just go around killing people to try to see if your medicine works, right? And you have to get their approval to use the medicine on them. And they have to be somebody who has the disease, right? So has the pneumonia and is in the hospital. So it's a pretty small group of people already, and they're already having a really hard time, right? So this would be pretty hard to test. Just throwing that out there. And maybe if they're super critical people, that's possible that they would, as Colin said, be taking the risk because maybe it's that or they're going to die, right? In which case, if you tell people you're either going to die or you're going to try this thing, they're probably going to try it, right? But they might not. They might say, no, whatever. I'm 98 years old. My time is done, right? You never know. They might refuse for other reasons. So it does get pretty complicated. Now, let's see. One way they used math. So in this paper, really all I did was summarize the abstract and some of these results. So this page right here, page 4, is where we start to see some graphs. Now guys, graphs count as math. Who knew? Right, but here we're actually looking at results from their experiment. So what did they actually do to prove that this interferon was important for having the body's cells be recruited or to have the body's cells go into action? How did they test this? This is a pretty hard question. I did talk about it in my 45-minute read-along. Yeah, they tested this on mice, right? And why do you think we would use rats/mice, right? Same thing. Why would we use mice? They're cheaper and it's legal to kill them, basically, yeah, right? They don't sue, yeah. No mouse is coming back to you saying, you gave me some weird experimental drug, I'm suing you, yeah. So it is sad, right? I know a lot of people say, oh, blah, 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 blah. Most of these mice are actually bred specifically to be used in experiments, or they, for whatever reason, are captured from somewhere because there's too many mice or whatever. It's mostly ethically obtained mice. I will say that, right? They don't just go out on the street and grab a mouse and just do whatever they want to it. But mice share a lot of DNA with you, right? They have a ton of DNA in common with you. In fact, a lot of what we know about how genes and DNA work comes from our understanding of mice and how mice is that even the right word? Yeah, I guess it is. The mice genes work. There we go. Sorry, I went on a field trip today, so my brain is not here. So they used mice, and what they did was they had 2 groups. They had one group of mice who they didn't do anything to. Those are called the wild type, right? Wild as in nothing was adjusted on them. The other group of mice, they knocked out the gene that produces that interferon molecule. They do this with, I know it sounds insane, but it's true, a gene gun. They literally shoot the gene and break it so that it can't work. So the experimental mice, they were not able to produce the interferon molecule. And remember, that interferon molecule tells the body's cells to kill so that you can get rid of the infection. So what we would expect to see is that the wild type mice that we didn't do anything to, they're going to survive more, right? Because they're able to fight the infection. So then we actually look at these graphs and we start to try to break down what they actually did here, right? Oh, and then by the way, both groups of mice, they then injected with pneumonia, right? Because they got to get pneumonia. So looking at this first graph, we see percent of survival, days PI, that means days post-infection. So our wild type mice are the dots, and our mice where they had their gene knocked out are the squares. So notice that in the beginning, all the mice were surviving. Big surprise, right? Day 4.5 is where we start to see, days 4 and 4.5 is where we start to see some changes. The mice who didn't have interferons, they started dying off pretty quickly. Around day 4, we go below 100%. Then we go down even more, to where at the very end of 10 days after getting the pneumonia, less than 50% of those mice ended up surviving. The wild type mice, the mice where we didn't touch their genes, they were able to produce the interferon molecules. Almost 100% of them survived, right? Maybe 90%. That's pretty good data, right? Of course, then they did statistical tests to prove that this was a big difference, right? Because just looking at this graph maybe is not enough to tell that. Over here, we also see a graph about body weight, where they actually show that the sick mice also lost a ton of weight, right? So they weren't able to eat. They essentially starved to death, a lot of them, which is often what happens with lung infections. Then they did more analysis on different parts of the body, right? The spleen and the liver. They also took the lungs, they took slices of lung tissue and analyzed how much infection was present in the lungs. They looked at inflammation, they looked at how much bacteria was there, how many immune system cells were there, right? And they sort of compared those things between the two different types of mice, right? And then if you keep going through the paper, you'll see there's just endless pages of data. Now, in the very bottom of any page with data, they explain sort of what they did, right? So wild type and the mice that were missing the gene, there were 12 of them in each group. They were infected with the pneumonia, right? And then they were monitored for 10 days. They used a Kaplan-Meier survival curve. Another statistics test, we didn't even talk about that, right? Their statistical evaluation, they used a Mantel-Cox test, also known as a log rank test. That's another test that we didn't talk about, but it's a test. They used weight changes, right? They used an unpaired student's t-test. Now, that was the test that I told you guys we weren't going to do in this class because it's really hard. Well, that's the test that they used, right? So if you read through all of these descriptions of the data, you'll actually see which specific statistical tests they did, right? And this is so that you could check what they did, right? Where they literally will know from their statistics test, is my information significant? Because maybe 50% of the mice died, but maybe that's really not that significant, right? It's hard to tell without running a statistics test. So they did a ton of statistics, and in fact, at the very end of the article, you notice a million charts that I'm passing, right? We're not going to talk about all of them, don't worry. They have their discussion, blah, blah, blah. All right, here we go. Their methods and materials, right? They have an ethics statement. This was done in England, right? So they have like different laws about stuff, but regardless, they've mentioned all the laws that they followed, right? That they obtained the animals ethically, blah, blah, blah. They talk about how they took care of the mice. They talk about how they grew the bacteria, how they cultured the cells, how they isolated things, how they modeled this, right? Literally, you have to break all of this down in a science paper. Now, how many years do you think it took this team of scientists to produce this paper? Five is a good guess, Colin. That's pretty accurate, right? It might be more. Usually when you do a study like this, if you're in graduate school, this will be your entire time in graduate school. You'll spend 5 to 7 years on one experiment, one, right? That's how long it takes to do this, because notice how much detail there is here. They had to break everything down from where they got the bacteria to where they got this to where they got that, right? Every single detail is covered. Now, the hard part is, imagine that you gave this to your parents and said, hey, what do you think about this study? right? Would your parents be like, oh, let's talk about the immunoblot analysis and the way that they did the enzyme linked immuno. My mom would be like, what's this, right? She would not understand any of the science here because she's not a scientist, right? And she didn't have anyone explain it to her. So the problem is a lot of the information we get about science studies, somebody has broken it down to explain it, right? And we have to trust that that person did the right stuff, right? Like I broke this down and explained it to you. You have to trust that I explained it correctly, right? What if I didn't? It would be really easy for me to lie, right? It would be super easy for me to look at one of these graphs and say, oh yeah, their paired students t-test just shows actually that it doesn't matter. All the mice died, right? And if you didn't know better, you would go, oh, okay, I guess it doesn't matter, right? So basically what I'm trying to say is always be critical. And look at the source yourself if you can. Even if you weren't able to understand every single word from this article, I bet even from the abstract, you were able to get a couple things out of it, right? So that's important here. There are lies about science on the internet, right? There's tons. In fact, one of our virtual sessions in a few weeks, we're going to talk about a thing that people were saying was showing happiness and all this stuff. What it wasn't, right? So we're going to talk about that a little bit. But basically, that is why it's hard to understand some of these science things, is because this is what they produce from a science experiment. And sure, it's open access, right? Not all of these studies are. A lot of them you have to pay to read, first of all, which then, of course, no one's going to pay, right? Especially if they're not a scientist. But also we're relying on other people to break this information down for us, which can be hard. Okay, sorry. I have to like rant about that because, you know, people take science for granted. They think everything they hear is true, and it's definitely not. Okay, so back to the questions. So that was the math, right? They used a lot of statistical tests, essentially, is what you should have gotten out of that. You can look at those graphs. I encourage you to look at one graph and try to understand it. Read the axes, see what they did, right? See what you think happened there. Okay, so we already talked about why hospital pneumonia is so bad. So we're going to answer questions 6 and 7 here, and then we're going to be done with this virtual session. So question 6 is, what is one way that reading this paper or listening to me talk about the paper has strengthened your understanding of science? It shows how hard science is and how difficult it is to put together a coherent study. Yeah, honestly, Joelle, that's a good conclusion to get out of this, right? There's a lot that goes into discovering this stuff. It's not just something that you can do really easily. It helped me to understand the way our body's immune system works to fight diseases, and it strengthened my understanding of how research studies are made. Yeah, right? So when we get frustrated sometimes thinking, well, why can't they just make a cure for this? This isn't even getting to the stage of making a cure yet, right? But just think about how many of these studies there have to be before we can even move on to making this a medicine, right? So it's a really slow process. And I'm not saying that's a good thing necessarily, but it is slow. So Joelle and Colin both gave great answers there. Let's see. Oh, we got one more. Reading this paper helped me understand how much work goes into the medicine field. Yeah. And how they produce antibiotics. Yeah, well, so they didn't really talk about producing them necessarily here, but producing alternatives to antibiotics. And then final question, an opportunity to get some points. What was the hardest part about reading this paper? Colin was ready. Yeah, the vocabulary is complex and not easy to digest. It's true. Just when you think you've learned all the words you need to learn, they put another word in, right? And you're like, what is this one? It's frustrating, right? It's hard. Even I had to read really slowly, break a lot of things down, look things up, right? It was definitely hard vocab, for sure. What else is hard about it? Yeah. The vocabulary and how to put it together to form a sentence and then make sense of the sentence. The graphs. Yeah, right? So we sometimes think graphs are easy. We're like, oh, I understand graphs. Well, they're not the easiest thing to understand sometimes. You really have to look at those. And if you don't like math or graphs, then you definitely were turned off by those for sure. Any other thoughts or insight into what was hard about this? What about the length? I don't know if you guys noticed, but the entire paper was 29 pages long, right? I think out loud I read to you about a page and a half. The fact that you don't have a ton of background on immune system, that is an excellent, excellent point, Georgia, right? I tried to give you guys some background, but of course I could have given you 7 lessons of background and it still would have been really hard to understand. Hopefully you know a little bit more about natural killer cells and macrophages though now. Oh, nice, Briley. I agree with Colin. The vocabulary was very complex and hard to figure out what it meant. Someone giving you a point for speaking to another person in the class here, adding on to what they said. Okay, so what you're going to turn in is your answers to these 7 questions. You're welcome to use things that were mentioned in this virtual session, right? If you didn't get a chance to yet, go watch the video I made or read the article on your own, right? Looking for answers to these questions. The background information, right? Read through the PowerPoint again about the macrophages and the natural killer cells. We will touch more on immune system stuff and antibiotics in semester 2. But I picked this article out of a few that I looked at just because I thought it was actually super interesting that somebody is finally thinking about using our own body as a solution. I think it's amazing. I've never seen that in all the years I've studied science. So it's pretty impressive. Also, I really wanted you guys to see what real science is like, right? A lot of this class is about that. A null hypothesis, statistics, right? These are things that real scientists deal with every day. They didn't even really have a null hypothesis explicitly stated here, but they do sort of hint at it, right? They're assuming that the interferon doesn't do anything. And then in reality it does, which is great because that's what they put all their money into investigating. Okay, Bonnie already sent hers in, good. So here's all the people that I have who spoke. I have Joelle spoke, Abigail spoke, Briley spoke, Georgia, Bonnie, Colin, and I wrote Georgia twice for some reason. And then these are the people that I have adding on to what someone else said, Briley and Georgia. If I missed you, please let me know. Otherwise, in our next virtual session, next Tuesday, we will discuss our final discussion on statistics. We're going to talk about averages. Right, and the problem with averages, really, and why they can be misleading. So we're going to talk about the mean, as it is mathematically called. So you don't have to do any pre-work for that. In fact, I'm going to rewrite the PowerPoint so that we can use Nearpod because I want you guys to be able to draw. So I have to do that by next week. So next week, you're just going to come and participate in the session. Otherwise, if you haven't turned in your pre-work, make sure that you do that now, and I'll be grading you on the rubric. And otherwise, we're done. So thanks for listening to me rant about science, as I like to say, because it's been about 45 minutes. A lot of it was me talking. Great job for everyone who participated, even if it was something that you were like, I barely understand this, right? You guys all pushed yourself to do that. So I will see you next Tuesday, and I will see some of you in Avid tomorrow.